Let's move on to Jack's talk. I'm very excited to see what he's gonna show us today. He's got some videos, I believe.
Yeah, it's the same old stuff. Nico had a whole set of great new slides I haven't seen before, in case you didn't realize that. He went to a huge amount of work, and I did exactly the opposite. I have almost all old slides, because I'm making a point that I've made many times in the past. I'm going to talk in 30 minutes about Voxel-Wise modeling, why it's awesome, why you should try to do it, why it's way simpler than you think, and why it replaces everything else. My usual position. So really, if you think about what we want to do for data analysis, you know, how do we want to basically design our MRI data processing pipeline? We want a method to test a bunch of different hypotheses all at once, hopefully on the same data set, because MRI is really expensive and time-consuming to collect data. We want to get very rich behavioral and brain data for as cheap as possible. So we'd like this to work on a small number of subjects. We don't want to record from 25 subjects if we don't have to. We want every assumption in the entire pipeline and modeling to be completely explicit and quantitatively stated so that there's no hidden assumptions. And in particular, all the operational definitions have to be explicit. This is one of the horrible, ugly truths of psychology, is that most psychology phenomena are filtered through an operational definition, and many of the assumptions of the operational definition are not stated explicitly, and it really holds our field back. We want easy bridges between psychological concepts and brain measurements. We want to recover fine detail in cortical maps, meaning all the detail that is there. In current MRI, that detail is not great, but we want to recover whatever is there. We want to minimize type 1 error, of course, everybody's concerned about that, but also we want to minimize type 2 error, which is really the main problem with MRI, is that basically it's rife with type 2 error. Most of this stuff in MRI, most of the stuff in the brain you cannot see in MRI. And we want to provide an objective measure of significance and effect size and prediction accuracy. Basically, we don't really want statistical significance, but we want to measure importance. So let's go take a step back here and look at some data for a second and think about how we want to do this. Okay, so on the left, on the bottom here, great, a green laser pointer. On the bottom is a movie people watched in an MRI experiment. They were just fixating on a little tiny dot you can't see here. And on the right is our MRI data. And as you all know, the brain's inconveniently folded up inside the skull, so it's nice to inflate it and flatten it out. And red here just means more bold activity, blue means less bold activity relative to the mean, and you know, these patterns are complicated. There's about 100,000 voxels in cortex. The voxels in this particular experiment, the TR is 2 seconds, but that's fine. There's very little data in MRI signals below about 0.3 hertz anyway. You can see painted on here are the individual ROIs we know about from other localizer experiments, and you can see that these patterns are pretty complicated. And what we would like is a method that allows us not to just find out any individual information about an ROI, especially binary information like animate versus inanimate, but we'd like a method that will allow us to understand this entire pattern of activity across the entire cortex, to divide up the cortex into functionally interesting regions of interest, to understand what the representation of information is in each of those regions, and to build a model such that for any given location we record, we can completely predict what that voxel does under any possible situation. And finally, just for an application, we might want to decode. So we want a method that will allow us to do all of that. So the way I tend to think about this problem, but you don't have to, it's just one convenient way to think about it, is what you want to do is find a linearizing feature space. So if we start with a very complicated stimulus, like I showed you in this movie, we have a bunch of pixels, and we have a bunch of brain activity, and we want to find the mapping between these. And one way you can always find a mapping, assuming your system is stationary, let's just assume that for the time being, is you can always find a feature space such that the mapping from the pixel space to the feature space is nonlinear, and the mapping from the feature space to the activity space is linear. And you can always do this because you can always stick a linear system at the end of any arbitrary system and it doesn't change the system. So these nonlinear transformations into some feature space are essentially a bunch of hypotheses that you can test, and this is just linear regression. So we're going to divide up this problem of mapping these pixels onto these brain data into two parts, a theory part, which is a hypothesis, where we're going to build a mathematical model, and a linear regression part, because linear regression is something we can do really, really well. So in this Voxel-Wise modeling framework, we're going to optimize our experimental design, pre-processing and statistical analysis to minimize type 1 and type 2 error. We're especially concerned with type 2 error. We're going to use 2 different data sets, an estimation set, which we're going to use for fitting data, and a validation set, which we're going to use for testing data. And really, there's only 2 differences between the Voxel-Wise modeling approach and what everybody else does. One of them is we always collect 2 data sets. And the reason for that is, as Nico mentioned earlier, to prevent overfitting. But we're not only going to collect 2 data sets for decoding, we're going to collect 2 data sets for everything. And secondly, and then we want to focus on prediction accuracy rather than significance, although we'll get significance for free. We want to avoid arbitrary decisions about data processing and any arbitrary suboptimal decisions, in particular smoothing decisions. Smoothing data is a regularization procedure. There is an optimal regularization to be done, but if you pick an arbitrary smoothing kernel, like a 6 millimeter kernel, an 8 millimeter kernel, that is not optimal, and we don't want to make any decisions that aren't optimal. We want to model the system in terms of low order models, as low order as we can. So for example, if we're measuring covariances, we want to measure the variances, the univariate models first, and subtract them off and measure the covariances on the residuals. We don't want to average data across subjects until the end. We want to analyze as much as we can in individual subjects. And then we want to interpret the results in terms of prediction accuracy, Voxel-Wise receptive fields, feature spaces, and cortical maps. So this is a little illustration I made. I'm not happy with this slide, but it's the best I can do right now. Imagine I have some big long movie. I can divide the movie into an estimation set and a validation set, 2 parts of the movie. And now I can just interleave these estimation videos and these validation videos. And once I create this interleaved data set, and interleaving is important because remember the system is non-stationary and we're going to pre-process it. So we want to make sure that the estimation and the validation over time get pre-processed as similarly as possible. Now we collect some MRI data, so now we're going to get some bold signals. The red ones are the estimation signals, the blue ones are the validation signals. Now we're going to pre-process that data. How we decide to pre-process it, of course, matters, but for the purpose of this talk, we won't cover that in detail. Now we've got some pre-processed bold data. In MRI, this usually means we speared our data. We started with non-Gaussian data, we ended up with Gaussian data. We don't know what the right way to process the data is, we just make it Gaussian. That's what everybody does. Now we break the data up into 2 sets, our pre-processed estimation data and our pre-processed validation data. Now in the experiments in my lab, the estimation data are almost always just run once each. So every one of these estimation movies, we only have 1 response to it. And the reason for that is when we fit our model, we want to sample as many different videos as possible. But the validation data, those are repeated. And the reason for that is we're going to predict in this validation set. And you can't predict a single trial, well, not well, but you can predict a mean. So by collecting this validation data set over multiple repetitions, we can calculate the mean response to this validation data. And that mean comes along with a standard deviation, and that comes along with a standard error, and that allows us to calculate the noise ceiling. So without having any model, we can say, how much of the variance in this validation data set would the best possible model predict? Say it's 70% of the variance. Okay, well we know that's the noise ceiling. That's what we want to hit, 70% of the variance. Okay. Now, we set this validation data aside and we do not look at it. For those of you, none of you are old enough, never mind. I was going to mention Al Gore's lockbox, but nobody remembers that joke anymore. We're going to set this aside somewhere and not look at it. We're going to take our validation data, our estimation data, and we're going to use GLM. Why GLM? Well, really, in statistics, there's only 2 things you can do. You can do regression or you can do classification, and those are the same thing, so really there's only 1 thing you can do, so everything's going to devolve to the GLM anyway. So you use the GLM, and you get a bunch of betas from your GLM, right, for your model, in whatever feature space you have. Now, you take those betas, and you use them to predict the validation data, and you get some prediction. If the noise ceiling was 70% of the variance, your prediction is 50% of the variance, you know exactly how good you're doing. You are doing 5/7. You're 5/7 to the way done, right? Now you might say, well, wait a minute, I don't have this weird, stupid movie experiment. I have some standard experiment. Like, you know, in one condition, people are experiencing pain, and in the other condition, they're not. And I just have 2 conditions, and this just seems kind of stupid and weird. But remember, if your data are analyzed by a t-test, as most MRI data are, a t-test is just a shorthand for regression, right? A t-test is a discrete regression fit with OLS, and there's just a shortcut that allows you to avoid doing the regression. If you chose to be a masochist, you could solve your t-test by regression. And in that case, you can fit your t values here and your betas that you need and your two conditions here, and you can predict here. So you can still implement your normal data processing pipeline in two different data sets. The problem is everything up to here is consistent with FSL and SPM, and this step isn't. You can't actually take the, well, I mean, you can, but it's not a standard thing that FSL and SPM is going to let you do, but you can predict on a separate data set. You might have to do a little bit of coding by itself. Now, why do you really want to do this? This keeps you overfitting to noise, obviously, because you're going to predict data that has different noise characteristics than the data you fit. So that's the number one thing you want in data analysis is do not fit to noise. The second thing is, though, these data don't have to be the same as these data. You could actually have different movies here than were here, and so you can predict outside of set, and that allows you to generalize your results, which is a critically important thing. So here's our data processing pipeline in the lab. First, we collect some bold data. There's a bunch of steps in that, right? But these are all kind of normal steps, except we tend to be really neurotic. But other than that, these are normal steps. We pre-process the bold data. That's the same process that everybody has to do when they pre-process bold data, correcting for motion artifacts, aligning brains, removing drift. Then we specify a model framework. A model framework is essentially a linearizing feature space. So we're projecting our stimuli into a linearizing feature space. If you have an experiment where the conditions are determined beforehand, then those conditions determine the linearizing feature space. Now we fit the model to each and every voxel, and we fit each and every voxel individually. We haven't smoothed the data, so each voxel is just dealing with whatever noise it has. Every voxel is going to get fit individually, so we're going to end up with a GLM for every one of 100,000 cortical voxels. Now, we can do this a bunch of times using different models. Eventually, you're going to get bored of this, and so then you're going to just declare victory. And when you declare victory, I should point out that this step are both using all the data, but the specifying the model framework, well, that's not a data step. But this fitting models to each voxel, this gets done only in the fit set of data. The test set is preserved. And after you spend forever exploring the fit set and fitting whatever kind of models you want, then you can unmask the test set and get your prediction on the test data. Now, of course, when you're fitting models in regression, you oftentimes need like a regularization set, and you need a model fitting set, and then you want a prediction set. So you actually take your fit data and you divide it into subsets, where you predict data inside your fit data. So you're treating your fit data as test data. But you don't touch the test data until the end. Now, eventually, you're going to unmask your test data. You're going to take the models you fit in this step, and you're going to get predictions. And then you'll either be happy or sad, depending on what happened. So now you can visualize the data within subjects by, say, plotting individual voxel receptive fields, which is just projecting the models into some visualization stage. Or you can visualize the data across subjects. That is a very tricky thing to do. We have a new framework, a generative modeling framework called Pragmatic for doing this, but I'm not going to talk about that today. And you can do decoding. Now, most people, when they're doing decoding in MRI, it's essentially symmetric with linear discriminant analysis. It's really an encoding question they're asking. They're asking, are two groups different? They're not trying to reconstruct a stimulus. But if you want to actually reconstruct a stimulus, which is the engineering application of decoding, you can do that. Hey, Nico, what time is it? Oh, wait, I've got my clock. Oh, I'm okay. All right. So this is an illustration, again, of how we do this. Here's our fit data set. So we have some movies. We have some MRI data. We hold the MRI data for a second. We take our movies. We filter them into different feature spaces, like maybe a motion energy pyramid, or we might extract the surfaces if we think there's some intermediate surface representation, or we can extract the semantic content from the movies. And now for each and every voxel, every one of our 100,000 voxels, we fit all of these models. So to do that, we take the weights from the individual feature channels, and we essentially use ridge regression to find a set of weights that map that feature channel onto the bold responses. In a typical MRI data, you usually have enough data, believe it or not, in one MRI voxel to fit, say, 6, 8, 10,000 parameters, something like that. So you can divide these models, you can have as many models as you want divided up among those 6,000 or 8,000 parameters. Right? So a typical motion energy model might be, say, between 2,000 to 6,000 parameters. Now, you take your other data set, your test data set, which is the validation data. This uses different stimuli, but it doesn't have to. If you have a low-dimensional classical experiment like pain versus no pain, then the conditions can be exactly the same. You just won't be able to generalize out of set. And now you take your test data, you map them again through the feature spaces, and now you multiply them times the beta weights that you estimated in this previous step. And now you take your bold responses and you just compare the observed bold responses to the predicted bold responses, and you use the predictions to determine which of these models each voxel prefers. So now, in this paradigm, every single voxel tells you what hypothesis is most appropriate for that voxel. And some voxels may like more than one model, or the models may be collinear, but that's just life. Now, eventually, you'll get tired of playing this game, and you'll have some models that you think are good enough or as good as your data is going to support, and you'll decide it's time to interpret your data. And there's kind of 3 methods you can use to interpret the data. One is you can pretend a voxel is like a neuron, and you can look at its receptive field. That's called a Voxel-Wise receptive field, or in a special case, a population receptive field. The second thing you can do is you can try to find a space analogous to what Nico was talking about, his dissimilarity space, that organizes all of the features sort of conceptually in terms of their similarity. And you can also find how that space is represented across the surface of cortex. So in the time remaining, I'll go through a few examples of these, and then the talk will be over. So here's a motion energy model. This is from Shinji Nishimoto's movie reconstruction paper from 2011. We had some movies, and we had some bold responses. So this black square, indicates one voxel. We want to model this voxel in terms of its responses to all the individual oriented motion energy in this movie. So we implement a set of motion energy filters that vary in terms of orientation, spatial frequency, position, and size. There are about 2,000 of them in this particular experiment. Now, for each individual motion energy filter, it's actually this cascade. So the movies come in, you strip off the color, you shove them through a Gabor wavelet, you take 2 of them in quadrature phase to make something that's kind of like a complex cell, and you stick a log nonlinearity in there. Why? Because it helps predictions. And now all of this happens at the movie rate, which was 15 hertz, and then you temporally downsample it to the MRI rate, which is 1/2 hertz. So now this gives you a filtered output. So now the movies get transformed through these filters to produce filtered output. This has nothing to do with the brain. This is a hypothesis about what the brain does. So to fit these features, for each individual one of these features, we fit a 4-tap finite impulse response filter such that the sum of these filters optimally produces the bold response. And this is essentially just going to be done with ridge regression. So this is a straightforward procedure, and it works pretty well. Now, once you fit a model, the very first thing you always do is you look at predictions. This is not a good slide because it doesn't tell you about the noise ceiling, and there are no numbers on it, but I have it here for a reason, if a bad reason. This is a flattened cortex. This is prefrontal cortex. This is the visual system on the left here. The areas at the back on the far left are the retinotopic areas, V1, V2, and V3. And around V1 and V2 and V3 are these higher order semantically selective areas. And blue here shows the value of the predictions of the structural model, the motion energy model. And you can see that this model fits pretty well in low-level visual cortex, exactly where you'd think. Now, you can look at, in the interest of time, I'm going to skip this. Okay, now if you want to look at a voxel receptive field, it's fairly straightforward. You have your voxel. Your voxel has a bunch of beta weights. Those beta weights are weights of features. So you take a weighted sum of features, and then you can marginalize out dimensions that you are interested in. For example, the model here, this motion energy model, is a pyramid. It's a spatial pyramid, so it has spatially localized filters. So you can recover the spatial receptive field of an individual voxel. Here's one voxel, and here's another voxel, purely from responses to movies. And on the right here, I'm showing retinotopic mapping. On the top is the retinotopic maps you obtain from movies, and on the bottom is the retinotopic maps you obtain from using a very sophisticated retinotopic mapping method. You can see these retinotopic maps are almost identical. The movies aren't quite as good, but if you think about it, in the movie case, we weren't optimizing at all to recover retinotopy, yet we still recover it in this model. Now there's a simplified version of Voxel-Wise receptive fields that you can use if all you care about is retinotopy. And this is a method developed by Serge Dumoulin and Brian Wandel, and a lot of people use it now. It's called population receptive fields. You take your normal wedges and rings, and then you use also bars at various orientations. And when you aggregate all these data together, and you assume that the visual receptive field is, say, a Gaussian blob, now you've got essentially a constrained regression problem, and you can shove the data through a normal regression procedure and find the optimal blob that explains the population receptive field data. So PRF and voxel-wise modeling are essentially the same thing, because this is also done on an individual voxel basis. The only real difference is that this is operating in a spatial subspace. Now, if you, here's just another example, which I'm introducing in order to address the cool things that Nico was talking about. This is a Voxel-Wise semantic model. So here we have a movie, and we go through the movie, and we use this giant hierarchy of inheritance called WordNet to extract all the nouns and verbs from the movie. In this case, not all the nouns and verbs. We classify the 1,785 most common nouns and verbs in the English language. We go through and we find them all in the movie. So now we've converted our movie into essentially an indicator matrix where the rows are the 1,785 nouns and verbs and the columns are time. And now we're going to perform regression again of this indicator matrix onto the bold data we get from an individual voxel, which is just a time series. And what we're going to get out of that is a finite impulse response filter that tells us how this individual voxel changes over time when some specific semantic event occurs in the movie. And we're going to get that for all 1,785 semantic categories simultaneously. And now we can look at the predictions of this model, and you see this model fits well in the semantic area, is exactly where you'd expect. And now you can look at an individual voxel receptive field. WordNet happens to be a tree, so it's convenient to show this as a tree. And essentially, in this tree, the size of these little circles are all semantic concepts living on the tree. The size of the circle indicates the beta weight, and the color indicates whether it's positive or negative relative to the mean. So you can see that this voxel gives a positive response whenever anything with a face occurs. These are all kinds of people, carnivores, ungulates, rodents, or anybody talking. And this is a voxel actually in FFA, so this is exactly what you want to see. Now, you do have a problem when you use this approach, which is you have way too much data, right? We have, say, 3 subjects. Each individual subject has, say, 60,000 voxels on the cortical surface, somewhere between 60 and 100. And we've had 985 semantic features that we fit to each individual person. And this is way too much data for any human to think about. So in this case, what you always do is the first thing you think about is dimensionality reduction. And the dumbest, stupidest dimensionality reduction you can do, which works well when all your data are Gaussian, which these are, is principal components analysis. So you do principal components analysis individually on each of the 3 subjects, and you find out it takes something on the order of 60 to 80 principal components to account for 95% of the data. So that's just a limitation of MRI. We have 985 regressors, but we really only need 60 or 80 PCs to account for these data. But now what we want to do is we want to find what of the subspace of this principal component space is in common or different across the subjects. So what you can do is you can take the data for subject 1 and subject 2, and you can correlate the principal components, and that's shown as a heat map here. So you can see the first 4 principal components for these 2 subjects are very highly correlated. For these 2 subjects, 2 different subjects, the first 2 are correlated. For these 2 subjects, the first 3 are correlated. Now, when you do this, you tend to find in most MRI experiments using complicated stimuli that you need something on the order of 3 to 5 principal components tend to be shared across the subjects. And the other principal components are essentially reflecting individual differences and noise. So once you find that the subject shares, say, 3 principal components, now you can just aggregate these three data matrices together, just make one big genetic matrix, perform principal components on that joint matrix, and take the first three principal components of that. And you end up finding that those first three, in this case four principal components, account for all of the subject's data quite well. So now you have transformed your data into this common space, this common underlying principal component space, which is a lower dimensional representation of the original features that you started with. Now you can do two things. You can take the principal components and you can project the features into this principal component space. Remember, there were 985 features, so we can look at how much each feature loads on each of these principal components. Or we can take the brain data and project each voxel into this principal component space and find out where in the principal component space each voxel lives. This space mediates between the feature space and the brain space in terms of a low-dimensional space. And this low-dimensional space is essentially, you can interpret as a similarity space. So here I'm showing you the first four principal components for this semantic experiment. Unfortunately, 4 PCs is inconvenient because my computer only has 3 colors. So let's take the first principal component, show up here at the left. Oh, my laser pointer is finally biting the dust. Okay, here's the first PC. It's up here at the left. And to make a long story short, this principal component distinguishes between bright, fast things and slow, dim things, which is not the most interesting result you could end up with. So let's ignore that one, and let's look at the other 3 principal components. So what we do is we take each individual concept, like this thing is some sort of carnivore, say it's a wolf, and we project this feature into this principal component space, where we're looking at the second, third, and fourth principal components, and we've colored each principal component on an RGB channel. And we can see here that this carnivore, if it's wolf assuming, is living somewhere down in this principal component space. Whereas if we look at this concept here, grassland, we can see that it's living up here in this principal component space. So all of the colors of the little circles on this diagram tell you where in this principal component space, which is shared across all of the subjects, each of the semantics concepts lives. And because these semantic concepts are projected into a principal component space, this diagram also tells you how similar the brain represents these semantic concepts across the brains of all the subjects. Concepts with similar colors are represented similarly in the brains of all the subjects. So all these yellow things are animals, and they're all represented the same in all the subjects. These green things here are people, they're all represented the same. These kind of brownish things are athletes, they're represented halfway between people and animals. These pink things down here are vehicles, these are indoor scenes, and so on. So this is essentially getting at the same kind of information that Nico was getting at with his analysis. Now, you might want to know about this, or you might not. You may want to know, say, how are these concepts represented in the brain? So now you take each individual voxel, and you map it into this principal component space, and you color it according to its loading on each of the 3 principal components. And then you map it back on the surface of cortex. So here we have a cortical map, right and left hemisphere, visual system in the middle, and we're mapping 1,785 colors on this map. That's obviously too much to think about. Let's just think of a few. So, for example, green is people. Anything you see in here is green is either people or body parts. Anything that's purple is vehicles, and so on. So you can find out how these concepts are represented. You can see that these data are extremely rich. Now, Nico and I love to argue with each other, and one of the arguments Nico has made is, well, wait a minute, but I don't care what the whole brain does. I want to know about what a local individual part does. That's one of your complaints, right? Yeah? Okay, he nodded yes, good. So how would you use this? How can we go beyond whole brain? Well, you can. You can take an ROI, or you can take an arbitrary region, and you can perform principal components just on that ROI, or just on that region. This is what my postdoc Tolga did with FFA. So he went into FFA, he performed the principal components analysis on FFA, and then he instituted, along with PCA, a cluster analysis. And what he discovered is that you can actually best cluster FFA, if you divide it into 3 separate functional clusters, each of which has different semantic tuning. And now you can take all of the voxels in each of these 3 clusters, and you can look at their tuning. And here's the tuning for the 3 clusters. You can see on the left, this cluster, which is in one, these are all spatially homogeneous, by the way. This cluster likes faces and all kinds of vehicles, and it doesn't like sort of abstract scenes and grass scenes and things like that, outdoor scenes. This second cluster likes faces. It doesn't really care about vehicles at all, and it doesn't like abstract scenes. And there's a third cluster that likes faces and doesn't like vehicles and doesn't like abstract scenes. These are 3 spatially distinct clusters in FFA. So the last thing I'll mention in the last few minutes I have is decoding. In the Voxel-Wise regression framework, you implement decoding as a Bayesian problem. The encoding framework nonlinearly maps some features into the bold responses through some linearizing feature space. And you can think of that as giving you the probability of a response given some function of the stimulus. And now if you want to get a decoder, which gives you some probability of some function of the stimulus given a response, you multiply this encoding model by prior, and you get a decoder. And you can think of that this way, where basically you're decoding, you're linearly mapping back into a feature space, usually weighting these bold responses by their likelihoods. And then if this nonlinearity is invertible, you can actually map back into the stimulus space. And this method produces by far the best MRI-based brain decoding anybody's done before. You guys have probably all seen this at this point. This is a movie on the left, on the upper left, and this is a decoded movie at the upper right. And this is a more recent algorithm that actually aligns the edges a little bit to give us slightly better decoding. And you can see that the decoding here is quite good. And this is only using this motion energy model from primary visual cortex. Now you can also decode the semantic model, which is only using that 1785 category model from higher order semantic cortex. And that'll start in just a second. In this case, we only decoded the 100 most frequent nouns and verbs because otherwise you couldn't read anything. And we're arraying them here on the first 2 principal components of the semantic space. So now we're decoding essentially higher vision. Here we get woman, man. There's no woman, but you know, the thing's not perfect. Hand, talking. There you can see the dog. We get dog, jumping, falling. This part is difficult to decode because it happens very, very fast. But now we're going to the ocean. You can see that we get sky, body of water. Now we switch to Waikiki, interestingly enough. So we get building, mountain, road, vegetation, walking, swimming. Now this part happens very fast, but we get text, you notice. Then the women come back. So we switch to getting talking, woman, man. This decoder works remarkably well. And there's no other method that exists in MRI that gives you decoding anywhere near this. And remember, the decoding here is essentially trivial because it's just Bayes' theorem. The reason this thing works so well is because the encoding models are preserving all of the information in the MRI data. They're not letting anything go to waste. And that means you have more signal, you have more signal to noise ratio, you have better signal, you have more variance you can explain, and you can build a better model. So I just want to flog our software for looking at these Voxel-Wise models. This is called PyCortex. You can see PyCortex in action if you go to my website, or if you go to NeuroVault and click on the PyCortex button or the 3-dimensional viewer button. NeuroVault actually now uses PyCortex to visualize the data. And you can download PyCortex from GitHub if you want and use it yourself if you're a really good JavaScript and Python programmer, because it's not really ready for civilians yet. And that's it. Thank you very much for your time.
Questions for Jack?
Let's just go to the common thing. And I think you and I are the only ones left. Is anybody else here? Did everybody else leave? Should we just yell at each other for the next 10 minutes?
That's a good idea. Yeah.
It's a good idea, exactly. Any questions? Questions? Or I can repeat it. I can repeat it. Whatever.
I would like to know how the receptive fields of one voxel was determined.
You want to know how they were computed? So remember, each individual voxel has a set of beta weights for its model, right? So say the model was a Gabor wavelet pyramid. A Gabor wavelet pyramid has a bunch of individual little Gabor functions, little oriented sine wave gratings that are localized, and they tile the spatial receptive field. So all you have to do is you can essentially, if all you cared about was the spatial receptive field, for example, you just take the individual Gabor wavelets and you sum them by their beta, you essentially multiply them by their beta weights and you sum that together, and that's the map you get. So that's always what you're going to do. You usually end up, because these are high dimensional spaces, you usually end up projecting the model into a lower dimensional space and then visualizing that marginal distribution. But you're essentially just looking at a map of the beta weights. Yeah.
Is this on? No.
It is now.
Okay, great. So my question is just, can you use the decoding model that you have with the concepts there? Have you guys tried using that with resting state data where, is that a possibility where we've always kind of wondered what people are thinking during the resting state?
Well, if you ask me about resting state, I can rant for about an hour and a half. The resting state, we haven't used this in resting state. Resting state is very difficult to do any kind of Voxel-Wise modeling. I mean, you can fit the Voxel-Wise model. The problem with resting state is when you have a fit set and a test set, those data are taken under a very non-stationary system, and that's being controlled by some state variable that you didn't measure. So the difference between fit data and test data in resting state is much larger than the difference between, say, a fit data and a test data if you're driving the system with some external stimulus, right? So that's why we haven't used this framework for resting state. As far as could you decode what people are thinking when they're resting, one observation I frequently make about resting state is that resting state overlaps enormously with the semantic network and with the activations you get during rumination. And I personally think that much of resting state data is people talking to themselves. And it certainly should be the case that you can decode internal speech from MRI data. I don't see any reason you can't do that. It's just a signal to noise issue.
Hi. So Voxel-Wise modeling is really great to study the functional properties of certain ROIs. But I'm wondering whether you have any comments about how you can use, in a clever way, these Voxel-Wise modeling approaches to study connectivity. Do you have any comments on that?
Well, I don't know if I can tell you a clever way, but I do have thoughts on it. So connectivity is just covariance, right? In MRI data, we take whatever data we get and we make it all Gaussian. And if all the data is Gaussian, then all the relationships between voxels are going to be covariances. So we're modeling a covariance matrix. There is no problem with modeling a covariances between voxels instead of voxels. It's just you're going to end up with the square of elements that you have to model. So you can model that using this framework. Again, it's just that your noise ceiling will be very low and your predictions won't be very good. You can also, however, model covariances in a task. So for example, you can have people watching movies and you can model the covariances between the voxels in that task state. The one thing I would recommend you be careful, though, if you're going to go down that road, is you really shouldn't be modeling the covariances initially. The first thing you should do is go in and model the individual voxels, and then take those predictions and subtract them off, and then take the residuals and model the covariances among the residuals. The reason is if you don't have a first-order model, if you don't have a univariate model, all the variance in the univariate model, when you build the covariance model, that covariance model will try to account for that variance too, even though by Occam's razor it should not. So I think when we go to the network modeling framework for Voxel-Wise modeling, we're going to want to build this up hierarchically. First you build the first-order models, and the second-order models, and then so on. And that's a well-posed statistical problem.
Another question coming out, possibly?
Yes, sir. Hello. Do you have an estimate? You have recorded functional imaging data in different spatial resolution using high-field scanners. And question is, do you have an estimate? What is the best resolution? Because of course it's a trade-off between noisy data and high-resolution data.
That's a very deep question. Here's the way I tend to think about it. I'm sure Nico has an opinion on this too. I tend to think of this in terms of mutual information. You have some task and stimulus variables that you manipulated. You have some brain data. You want to maximize the mutual information through that entire system. And oftentimes, for example, if all you want to do is get significance, you want to make the voxels as big as possible because you'll have more signal to noise, and so it'll increase significance. But if you think of it in terms of maximizing the mutual information in your whole experiment, larger voxels are not necessarily what you want. So, it's a tricky and complicated issue, and it interacts with all of the other things you did in your experiment. How did you do your pre-processing? Did your subjects move or did they not move? Are you using a 12-channel coil or a 36-channel coil? Did your subjects have a big head or a small head? So all those things matter.
And of course, another limitation is the amount of time you have for one subject to show images. And the question I have is, how have you considered, or it would be a great feature to measure a bunch of subjects, hyperline them due to one common movie, and then expand to other movies, and then build a database of which you can kind of combine between many subjects and build up, therefore, more and more data space. Does that make sense to you? Have you?
It makes perfect sense, and we're doing that slowly. I think when the Nishimoto paper was published, we had maybe 3 subjects. Usually that's the minimum I try to get away with. And now we've probably got like 15. And I'm trying to get money from NIH to collect 50. But we don't only want to collect 50 subjects worth of movie data. We want story data. We want resting state data. We want every localizer anybody's ever invented, like 5 or 6 hours of localizers. We want diffusion imaging, all that data set from individual subjects. And then we want to build one gigantic database. So in the part I didn't talk about today is this pragmatic algorithm, which is a generative algorithm for combining data across subjects without having to do any explicit hyperalignment. And that I think will be the direction in the future. We should talk later about this. Yeah.
More questions?
More questions. So what do you think about resolution? Because the resolution for similarity analysis matters, right? It must influence what you get in a representational similarity analysis.
Yeah, we definitely agree we want very rich stimulus sets, and we want the richest possible responses we can get. So, yes, we want to optimize the information about brain representations we get with our measurement. And it's a very interesting empirical question that there should be more studies on what resolution is best. For example, if you do something very simple-minded, you just try to decode and then try that at different field strengths and with different resolutions, decode something that's of interest as a kind of stand-in for more complicated questions, perhaps in a brain region like V1 where you have some idea of what's going on, and that would be very useful as a benchmark kind of for what kinds of acquisition schemes we want to be using.
So I want to comment on that, if possible, about the resolution. So actually, Dennis Chaimov and I are completing a paper, manuscript, about what is the optimal resolution in order to do decoding or reconstruct a functional map. And I'm talking about columnar resolution. And I believe that the question that is being asked here, although it's not necessarily about columnar resolution, is very similar. And what we find is that for reconstructing a map, or let's start from decoding. In order to decode optimally ocular dominance, orientation in V1, et cetera, under relatively low signal-to-noise ratio, you want to maximize your signal-to-noise ratio, and so the best way to do that is simply the sampling theorem, meaning for ocular dominance, get that in humans are about 2 millimeter per cycle. Voxels that are optimal for decoding are 1 millimeter wide. In order to reconstruct optimally the map, really to get, to present the map, and of course we're doing simulations so we know what the map is, you get just a little bit smaller voxels. Again, under the assumption of limited signal-to-noise ratio. So I think it's very relevant, and I'm saying that we're doing simulations, but actually I have data also both from humans and animals. Animals is optical imaging, humans functional MRI, to show that this is correct.
Well, I'm personally glad you brought that up, because the V1's a great example, right? V1, a neuron in V1 is coding in a 15-dimensional space, right? It's eye of origin, XY position in the visual field, spatial frequency, temporal frequency, direction, end stopping, right? You add all this up, it's a very high-dimensional space. And in MRI, at normal 3 Tesla MRI, we're using like a 2 or 3 millimeter voxel. We're taking a pretty big voxel that is actually averaging over a whole bunch of hypercolumns. And those hypercolumns are important because this high-dimensional space gets projected onto a 2-dimensional sheet. And the global variables are retinotopy, but the local variables are the interesting ones, right? And we can't currently really recover any of that local information. We're only recovering retinotopy and anything that's correlated with cortical magnification factor. So in animals, of course, there's a lot of ways to do this. In humans, there isn't, but there is one, at least, brains grant on that Larry Wald's involved in, David Feinberg at Berkeley and Chun-Li Liu at Duke, where they're essentially trying to build the next generation of coils. These are 256-channel coils. And the theory is that that will get us down to about 500 microns for functional imaging. And at 500 microns, that would be the scale that you would want, right? So if we can really get 500 micron data, I think it will really, it'll make functional MRI much more like neurobiology, much more like neurophysiology.
Maybe I've got a final question for you, which is, you haven't really talked about what the goal of all of this is, right? I mean, it's sort of a cool way of visualizing a lot of information on the brain. There is this element of having different computational models, and perhaps some of them work better than others. But what is the goal? Is it adjudicating between the models? Is it visualizing a semantic space?
It's all of those things. Oh, I'm not. It's all of those things, right? I mean, I don't know. I haven't ever.
I think so. In order to make all the decisions along the way, I think it's good to have one particular goal in mind, right? So, for example, if it is testing the different computational models, then it's interesting to think about this regression step. You said, so this is the theory step, and then we just do linear regression. But there's assumptions coming in at the step of linear regression that might affect which theory you choose, right? Similarly with, if the goal is to create a visualization, it would be good to know exactly sort of what is it supposed to show you, because otherwise you've got this sort of mass of a very complicated analysis between the map you're looking at and your interpretation of it.
But everyone has that problem. That's why you did a paper that compared, 15 different kinds of essentially correlation metrics in the context of the question you were trying to ask, right? All of these procedures involve assumptions. However, the nice thing about equations is the assumptions are, kind of well known, right? I mean, if it's a GLM, you know what those equations are. They're well understood assumptions. If you're regularizing and you're fitting a regularization parameter, you know what you're doing when you do that, right? So I think this approach and your approach, since they're in many ways similar, it avoids problems of, it basically takes the work, the information-bearing work in your experiment, and it moves it from this operational definition, this kind of vague conception you had about your experiment, into the data analysis pipeline, where it should be, where it's very clear. So in psychology, the way I was trained, the way all psychologists are trained is, okay, say I'm interested in love. Love's a great topic. Let's study love. Okay, well, how do I study love? Well, what's love? I don't know. Let me make a questionnaire. Okay, that's love. Your answer to this questionnaire is now love, right? So it's a 0 to 100, so that's your love score. Okay, so now I'm going to do everything according to this questionnaire, right? But there's huge amounts of assumptions, completely unstated assumptions, about the relationship between this operational definition and the thing you actually cared about. And pushing down the data analysis pipeline to making explicit mathematical models is an attempt to try to take the guts of that and make it all explicit so that other people can look at it and decide whether those assumptions were valid or not. And this is a big deal for me because I teach undergrad statistics, and they have a really hard time disassociating the operational definition of the thing from the actual thing. It's very difficult for them. And I think anybody, when they're in a field for a long time, they kind of get used to what they're doing and they kind of forget about all the assumptions that are involved in it. It happens to all of us, it happens to me. And this is kind of a way to avoid that. Now, as far as what the ultimate goal is, my ultimate goal, Nico, is the same as yours. It's world domination, right? I mean, basically, I want to have a model that for every single voxel in the brain predicts exactly how that voxel will respond under any possible circumstance that could ever occur in the history of the world. And once you do that, then you're done. So then the question is just how do you want to get there? It's an impossible
I think that's
goal.
a good last word. So this concludes this session. Thanks very much for coming.